When you take a pill, where does it go? Most of the drugs that are prescribed to us will go to many places in the body, often where it's not needed. And we are on the cusp of a medical revolution where we want to deliver medicines exactly where they need to go, when they need to get there. But to do this, our lab takes inspiration from systems our body already knows.
COVID-19 vaccine has brought RNA therapies to the global stage. Decades of research made it possible to develop the vaccine at record speed. And now that the pandemic is over, there is interest in improving the process. And it is precisely the way vaccines or medicines in the form of DNA or RNA therapies are delivered to the body's cells that Maggie Holme wants to improve.
We can make nucleic acids, which means DNA or RNA molecules, and then we can design them to make, in theory, basically any protein that we want to, because it's like a recipe book for how the enzymes in the body build proteins. One of the amazing things about this is that you can target, theoretically, almost any disease in the body. The problem is that we need to be able to get these therapies to where they need to go.
To transport medicine and vaccines, so-called nanocarriers are needed. These typically use layers of molecules called lipids to enclose the DNA or RNA molecules on their journey to their target cells. These carriers can be designed in different shapes or sizes, depending on what they are intended to transport. However, these lab-made nanocarriers will deliver their cargo throughout the entire body, rather than to a specific area.
If you have a vaccine, it just needs to go into your arm and the immune cells in the body will find it. You want to treat, for example, heart disease or something in the brain, then you have to be able to tell these particles to go to those parts of the body.
The body has its own natural types of nanocarriers called extracellular vesicles. Extracellular vesicles can transport themselves to a specific location in the body, enter the cell, and deliver information, for example, instructions from DNA or RNA. And it is these particles that the research group wants to mimic.
So the overall idea is to figure out how to make nanocarriers that are more inspired by what we know the body can do well, and to figure out what we've made, and then to figure out if it does what we want to do. The kind of two ways we're approaching this is from designing and testing how they work in cells, and also from understanding what they're made of. So what does a lot of these particles look like, and how do they differ on the particle-to-particle level? Nanoparticles that are produced by the body, these so-called extracellular vesicles, they have tens of thousands of different lipids in them. We will not manage to recapitulate that in the lab, but what we can do is try and better understand what are the parts of these vesicles that are important for what we want to do, and then how do we then take them and use them in a synthetic vesicle.
Already measured 1 in mile for you today?
Yeah, there's my second one.
So here we are in the lab. This is where we do a lot of the lipid nanoparticle formulation, we call it, and also where we look at some of the properties of these particles on the individual particle scale. So we couple this, what we call structural characterization, with how things actually behave in cells. So there we will feed these particles to cells and see how the cells are able to take up the particles and then process the model drug that we've given them. And then we can start to understand how we can actually apply this to treat diseases.
The group's findings can then be used by other researchers to develop drugs and vaccines that can be delivered more effectively to their target cells, which could mean smaller doses and fewer side effects.
We hope that this information will be able to help patients and also to contribute to the fundamental understanding of how we can, in the future, design better drug delivery systems and also understand how the cells in our body work. It's super fun to try and look at stuff on the single particle level. Things go in and out of fashion in science like they do everywhere else, and lipids were very fashionable in the 80s, and now after COVID, lipids are really cool again. And so there's this huge kind of regeneration and being interested in how these particles look.